Hello, welcome everyone. My name is Marcelo Vasquez, and I am the radiobiology head section and scientist at the low dose research program here at Canadian Nuclear Laboratory, also known as CNL. On behalf of CNL and our supporting partners, the Atomic Energy of Canada Limited, Health Canada, and the Canadian Nuclear Safety Commission, thank you for joining us today. We are very excited to have Dr. Aaron Darcy presenting today. He's part of our low dose radiation webinar series, and we're very happy to have you today. And as you know, or maybe you don't know, but the goal of this webinar series is to help strengthen connections between the research community by engaging world-leading researchers pertaining to the effect of low dose of radiation from different sources. If you have joined us before, you'll know that at the end of Dr. Goodarzy's presentation entitled Fundamental Biology, Molecular Epidemiology of Repetitive Radon Inhalation at Doses Relevant to Canadian Populations, you will have an opportunity to ask questions. Please use our Q&A function in your screen to write in these questions. I will moderate the questions. So please write your inquiries in this website. Now, let me introduce today's team speaker. Dr. Goodarzy is the director of the Robson DNA Science Center at the University of Calgary's Charbonneau Cancer Institute. He obtained his PhD from the University of Calgary in 2005, studying the biochemistry of radiation-induced DNA damage and repair. Aaron then trained until 2010 as a postdoctoral scholar, studying the cellular effect of ionizing radiation exposures at the Genome Damage and Stability Center at the University of Sussex in the United Kingdom. In 2011, he opened an independent radiobiology laboratory at the University of Calgary's Cummins School of Medicine. And since 2014, Dr. Goodarzy has led the EVICT Radon National Study. It's a transdisciplinary national research project aimed at enabling research to understanding and engineer out lung cancer causing radon gas exposure from the Canadian residential environment. So it's a great pleasure to introduce Dr. Goodarzy. And Aaron, the floor is yours. Welcome.
Thank you so much, Marcelo. Give me 1 second. I'm going to share my slides.
Sure.
Can everybody see that?
Yes.
Great. So I am currently the director of the Robson DNA Science Center, which is based at the RNA Charbonneau Cancer Institute at the University of Calgary. One of the things that we focus on at the Robson DNA Science Center is really our DNA and how that's affected by a variety of factors from the environment. We have three core themes at the Robson DNA Science Center. Today, which I'm going to really focus in on, is environmental cancers, which involves a lot of cellular biology, molecular epidemiology. We also look for therapies for rare cancers. That involves a lot of structural biology, and living medicines, which involves CRISPR, genome engineering, and others. Today, of course, I'm going to focus in on radiation at low doses that we encounter in the environment and how that relates to cancers. Right now, I'm in the middle of the Environmental Cancer Research Hub, which was funded by CFI and other partners, which involves our environmental cancer research program here at the University of Calgary. And our transdisciplinary or multidisciplinary, interdisciplinary, whatever word you want to use, where we really bring together more than just biology, really everything you can think of in order to fully understand this problem. Within this, we also have our alpha radiobiology lab, alpha standing in this case for alpha particles, and our cancer metabolomics lab that you can see there partially under construction, and which really drives forward a lot of the research that I'm going to tell you about today involving so many people. So I'll start where we really, the heart and soul of the Robson DNA Science Center, which is, of course, DNA. When you're born, you hope your DNA looks like this, particularly if you're female, you have two X chromosomes, two of everything else. And that's relatively unscathed as we come into this life. But of course, over time, our DNA is subject to damage, a variety of different damage. I'm showing you six of the most common types here. The DNA double strand break, oxidized bases, base mismatches, single strand breaks, cross links of multiple types, which really modify our health over the long term, including risk of cancer and other things. Of course, radiation will produce nearly all of these in some way, shape, or form, and they're really important for the rest of what I'm going to tell you about. Our focus is really lung cancer. Here are five of the most common sources of lung cancer. Of course, the old favorites like tobacco. We also have combustion particulates, all that wildfire smoke, of course, industrial pollution, and lung inflammation, arsenic, depending on where you live in Canada. And then what the big focus is today is radioactive radon gas, which emits alpha particles, can directly ionize our DNA to produce lesions that can drive mutations, and that alters disease frequency. When you're thinking about environmental cancer risk, it's really important to understand for a person the likelihood that they're ever exposed. For example, we always joke about radon. You know, if you live in a tree house, you're not really going to be exposed to appreciable quantities of radon. You do need to be a house on the ground. But if you do live in a house that's built on the ground, it's a question of how much and for how long. So dosimetry, I'm going to talk a lot about that today. And then there's the modifiers, susceptibility, which is, of course, hardwired through our genetics, our ability to repair DNA. And then the modifiable behavioral factors such as diet, lifestyle, fitness, and occupation. You have to really understand all of these risk modifiers together with the fundamental mechanisms and, of course, the effect of genetics in order to understand what's going on. So our science in my lab really has three core themes. We focus in on radiation research. I've been a radiation researcher since the year 2000 when I joined it at a ripe age of 18. We also look at oxidative stress, which is also really important as part of the radiation conversation, but also a bit bigger than that. And then, of course, our radon biology, which we've been intensively doing for the past decade since 2014. A lot of what we do does look at the genetics of susceptibility. So I will come back to that a few times. Here, for example, is a list of SNPs, single nucleotide polymorphisms, that are known to be associated with lung cancer, both in tobacco smokers and people who don't smoke tobacco. And many of these are of interest to us because they represent DNA repair proteins and others. We put out a lot of papers. This is, for example, a sample over the past few years on DNA damage biology in collaboration with others, including Canadian nuclear laboratories. So thank you very much to our hosts for inviting me. But also we put out quite a bit on radon, and that spans many, many other different fields, including behavioral science, building science, and of course, epidemiology. And so that's a core part of our program, which bridges biology with basically everything else. So the Evict Radon National Study, funded by Canadian Cancer Society, Health Canada, CHR, and the Alberta Real Estate Foundation at this time, involves a lot of people from a lot of different places across the nation. A core part of the Evict Radon National Study is citizen scientist-based radon testing of houses. So people getting a radon test from a research program, deploying that in their houses, gathering that information basically themselves, getting those tests back to us, and then we can understand indoor air household radon, which really does mean alpha particle radiation exposure. Now, why is this important? It's important because Canada right now is in a lung cancer crisis. We have, according to the Canadian Cancer Statistics reports, amongst the highest incidence of lung cancer and mortality rates in the world. And if you reflect on the fact that we have among one of the world's lowest uses of tobacco at this time, and for some time already, what is going on? This really speaks to the many non-tobacco sources of lung cancer. Indeed, in our cancer clinics right now, if you look across the nation, it's relatively comparable. We have almost 30,000 cases of lung cancer being diagnosed each year. Of these, there are multiple different types. We're seeing the types most associated with tobacco, small cell lung cancer, actually continuing to drop for both males and females. On the other hand, the types of lung cancer associated with non-tobacco lung cancer, adenocarcinoma, for example, are actually flatlining or actually on the rise over the past few years, really speaking again to these non-tobacco sources of lung cancer that we're all being exposed to. Indeed, so much that 2 out of 5 lung cancer patients we see right now do not present with a tobacco-induced lung cancer. One patient has never smoked in their entire life ever, and the other patient in green there smoked so little or so long ago that we can't even see a tobacco-induced mutation signature in many of their tumors. So 40% of lung cancer cases are not due to tobacco. So what's going on there? This, of course, comes with an enormous economic burden. And if you look by stage from the lowest, which is where the cancer is very localized, easy to just cut out, so to speak, to stage 4, where it's spread everywhere, these come with huge costs. In fact, these are the staging of diagnosis, overwhelmingly, unfortunately, diagnosed at end stage for people who don't smoke tobacco. They range from anything from $15,000 a case at stage 1, 25,000 at stage 2, 137,000 at stage 3, to the whopping 330 odd thousand at stage 4 diagnosis. Multiply that by all those numbers, you're looking at $1 billion a year problem in Canada. Now, again, our incidence of this is on the rise. This was a review article I thought I'd bring to everybody's attention, published just a few months ago, where they reviewed sort of the global spectrum of non-tobacco lung cancer. And about 10 years ago, it was understood that this was the 7th leading cause of cancer death globally. That's now the 5th. Overall in the world. And radon is listed as the 2nd leading cause of all lung cancers, and the leading cause amongst non-tobacco lung cancer. So I thought, before I get much further, I'd set the stage by putting everybody on the same page with regard to what radon is and why we should be understanding it. If you look at the typical person's overall lifetime non-tobacco lung cancer risk, a lot of that does come from radiation. And if we look at doses of radiation that we all experience, a majority of a person's lifetime dose will come from inhaling radioactive radon gas, about half or less, again, depending on your health. I say that because the artificially sourced radiation part of this pie chart is overwhelmingly CT scanning for medical, and of course that is very health dependent. Then of course you have a proportion from buildings, meaning what the buildings are made out of, cosmic radiation, food, and other sources. But radon's the big one when we're thinking of lifetime radiation dose. It is alpha radiation, which is weakly penetrating but densely ionizing, quite different from beta and gamma rays. And it naturally arises in the ground. It's a uranium, it's in the uranium decay chain, uranium to thorium, thorium to radium to radon. All of those are solid until they hit radon. It enters soil gas, which of course drives itself to the surface of the earth and then is released. It's unstable, it has a half-life of only 3.8 days, and emits alpha particles. These move at 15,000 kilometers per second with enough energy to dent impact-resistant plastic. So imagine what that's doing to the DNA of the cells lining your lungs. Radon is very much a human-made problem because we have built buildings on the ground that reduce the dilution of radon that naturally occurs as it hits the surface and dilutes on the wind and the air to almost nothing. In our buildings, what happens is radon will enter either via water, through wells, through cracks in the foundation, where it will accumulate to relatively high levels, and in some cases exceptionally high levels. Of course, then humans will breathe that in. The natural rate of radon migration in soil gas from underground migration pathways to the surface is also modified by humans, and we can talk more about that later if you're interested. Radon, of course, being unstable with a half-life of 3.8 days, then transitions to its next radioisotope. In that case, it's radioactive polonium. That precipitates, essentially. It precipitates in air, attaching itself to dust particulates, which we can also breathe in, but then, of course, precipitates in our lungs. Very quickly, it becomes radioactive lead-210 with a half-life of 22.5 years. That will bioaccumulate, and I have evidence for that, and I'll show you that later on in this talk. For every single atom of radon that you inhale, you're going to have 4 alpha particles emitted until it becomes stable lead-206. I often talk to the general public about this, and I like to put everybody on the same page with regards to radioactivity measurement. Radon is measured in becquerels, which represent the number of radioactive events per second, so the number of alpha particles emitted per second. I always use the analogy of me sitting under an apple tree. The radon is the tree, the alpha particles are the apples, the becquerels are simply the number of apples falling from the tree at any given time. In biology, we often talk about the gray. Those are the apples that hit you, so it's the absorbed energy, and the sievert, which are the apples that hit you and hurt you, meaning they have a measurable biological effect. If we use an analogy of these apples, and when we set one apple as one dental X-ray, we multiply that by 10, we might have a chest X-ray. That's relatively benign doses of radiation. Multiplied by this, that's a CT scan, a heftier dose of radiation in that case. Take one CT scan, multiply it by this. Now, this is, for example, the amount of radiation coming out of the Fukushima reactor back in 2011 when we had the meltdown. Why am I telling you this? I'm going to bring back my Apple analogy in a few slides to radon gas to contextualize this for everybody of what's going on. In terms of the very rich history of radon, how do we know what we know? It's actually a Canadian story in origin. It was discovered at McGill University by Ernst Rutherford and originally termed the radium emanation because, of course, it emanated out of solid radioisotopes such as radium. He wrote in his book, which is actually sitting on my shelf here, thus there can be little doubt that the abnormal radioactivity observed in caves and cellars is due to a radioactive emanation present in the earth, which gradually diffuses to the surface and collects in places where the air is not disturbed. So he wrote that in the late 1800s, early 1900s, published in a book called Radioactivity, describing the modern Canadian, in fact global, radon problem very well. This needs to be acknowledged as a co-discovery with Harriet Brooks, his graduate student at the time, and I think once in the running to be on the Canadian $10 bill, trailblazing nuclear scientist. Cut ahead to, a few decades to World War II, Wilhelm Hupper, which was the founding director of the US National Cancer Institute, wrote a book called Occupational Tumors and Allied Diseases, where he was reviewing several centuries worth of data on disease outcomes in European miners. And in that book, he concluded that radon was responsible for killing about 50% of the men, often boys, as you can see in this picture, who went down these mines, often maybe, for example, at 13 to 15 years of age. And by their 20s and 30s, they're dying of lung cancer because of radon experienced in those mines. The wide dissemination of this knowledge was somewhat precluded by the Manhattan Project. They didn't really want too much negative said about radiation at the time. It's said that every disaster movie starts with the government ignoring a scientist, a very salient topic in the modern era. And of course, that is kind of what happened. Hooper went ignored on this issue and cut ahead to the 50s and 70s when the civil and military uranium mining needed for the civil and military nuclear programs produced basically the same outcome. And in Elliott Lake, Ontario, the uranium mines were eliciting lung cancers in an overwhelming number of their workers. In fact, by 1974, a Royal Commission concluded that these miners experienced double the expected rate of lung cancer. So this is the obelisk in Elliott Lake, upon which are the names inscribed of the again overwhelmingly men who succumbed to radon-induced lung cancer. This was not just a Canadian experience; it was happening in the States and, of course, in the over in Europe, including behind the Iron Curtain. It actually continued to happen behind the Iron Curtain after the 70s, all the way through to the 90s and the fall of the Berlin Wall. And so it's very interesting. You have two cohorts: the North American cohort, for whom exposure was more or less stopped in the 70s, and a German cohort for which exposure continued an additional 20-odd years, and both have been studied. At the same time, in the 70s, animal work was being done on radon, using radon chambers. A lot of the outcomes are summarized in this publication from Unskier in 2000, which you can review. I'm sure many of you are familiar with it already. I took some of the data for that, made this graph. They exposed the rats, in this case, to radon gas in these chambers at known quantities with cumulative exposures, as you can see there on the x-axis, and monitored who got a lung tumor. They did it at two dose rates. And what's quite striking is that the lower dose rate, in this case of roughly 12.5 millisieverts a week, actually produced far more tumors, with almost 70% or more of these rats developing a lung tumor following known radon exposure where the controls did not. Why the lower dose rate? From a biological perspective, dead cells can't become cancer. Too high radiation exposure simply would more likely to be killing the cells than altering them genetically to the point where they develop into a cancer. So that's an important theme as we go through this lecture. Now, let's leave the 70s and go to the 80s. You can kind of tell because of the hair in these pictures. Who's this man? This is Stanley Watrous and his young family. Stanley was a nuclear energy worker, like many of us. He went to work one day here. It's the nuclear power plant, I believe, in Limerick County, Pennsylvania. And something weird happened to him as he went into work because he set off the nuclear detectors, basically things that detect radioisotope contamination on clothing. They went off when Stanley came into work, didn't go off for anyone else. They didn't go off when he went home. They thought it was a glitch. They'd just been installed. But something was up because there was no actual nuclear material on site yet at the plant because it was still under construction. Further investigation determined that what was going on was every time he went home to this little house, that house was so full of radon, indeed 99,900 becquerels per cubic meter of radon, that the solid radioactive isotopes that decayed from radon, polonium, lead, etc., were pouring off his clothes at such high quantities every day. He was setting off those detectors. This really sparked the first understanding of residential radon exposure. And going back to my Apple analogy, take a CT scan and multiply it by this. That was the equivalent dose of radiation from the radioactive radon in Stanley's house. We've seen these numbers in Canada. This is a house not too far away from my office here, which is registered at 100,000 plus becquerels. It's a very unusual case. If you want to learn more about that, you can ask me in the Q&A section. In the years that followed throughout the 90s to the mid-2000s, large epidemiological studies were done, many of these. They're summarized in the WHO's handbook on indoor air radon. I'm taking here the data from the European pooled study, which concluded that you add 16% relative risk of lung cancer for every 100 becquerels of long-term radon exposure that you might encounter in the residential environment. This was reproduced in North America. This was reproduced in East Asia from a Chinese study, and is backed up very well by all of the knowledge we gained from the minor cohorts. Here's half a dozen of the minor cohorts, which found anything from a 5 to a 56% increase in relative risk per 100 becquerels per cubic meter of radon. This is some of the most consistent epidemiology that we've ever seen for an environmental source of cancer. In fact, it's just recently been confirmed yet again with a linear exposure response relationship in a publication you'll find in the British Medical Journal from a couple of years ago. And from Radiation Research Society Congress in 2023, we learned that the million-person study has also found more or less exactly the same thing. This is solid epidemiology. Indeed, IARC, the International Agency for Research on Cancer, classifies radon as being absolutely known to cause cancer in humans and animals. Of course, those rat experiments confirm that. And so what does it do? How does it impact our health? And in the next few slides, what I'm going to do is really articulate directly how radon is impacting our health. So why is it so cancer causing? Well, if we compare it to photonic radiation, X-rays, gamma rays, which again move through us relatively quickly with a lower ionization density, particle radiation, such as alpha particles from radon, move much more slowly. They're much more densely ionizing. And of course, that exerts very different effects upon our DNA. A lot of it is about clustering. So here I'm modeling cartoonishly an X-ray going through us. You've got 5 little chromosomes sitting in the cell. It sounds like a nursery rhyme. Send X-rays through them, you're going to get breaks. They're going to be quite widely spaced. And we essentially have evolved to repair this. We have multiple DNA repair mechanisms that essentially take those pieces of DNA, put them back together, and most of the time, there is no mutation that will happen. Now, on the other hand, clustered DNA damage from things like alpha particles or heavy ions will have a much more localized shredding effect. Here you can see the little brown chromosome didn't get hit whatsoever, where the other 3 colors in some cases got completely shredded, like the dark blue or the pale yellow. Articulating that in what's actually going on, you have a lot of DNA fragmentation at the site of the breaks. And you have a lot of breaks all in proximity with a lot of different types of DNA damage. Essentially, this is a real cluster of a lot of different now competing DNA repair pathways that have to deal with a lot of types of different DNA damage all in the same place. This may well be the most cancer-causing type of radiation that we're aware of, and it'll take a cell from the normal phenotype that I showed you at the very top of the hour to this cancer phenotype. This is a typical genotype of a cancer cell, which is essentially complete genetic chaos. more than two chromosomes, or sometimes no chromosomes if you look at number 13 there. And then monsters there at the bottom, you can't even recognize what they used to be. That's the genetic face of cancer. That is what radon is doing to our cells. High LET linear energy transfer versus low LET radiation has been known for a long time to have a very different dose relationship with excess cancer incidence. So here we have on each axis radiation dose on the X and cancer risk on the Y. Look at those curves, they're incredibly different. Relatively high dose of radiation there in total absorbed dose over time is not really moving that cancer needle all that much relative to the same dose of high linear energy transfer radiation, which really doesn't take much to have a much multiplied effect. Again, when I talk about this to the general public, I often use analogies. And the great analogy I use, if you've seen Star Wars, is low LET is more like that laser that misses most of the time. It's missing your DNA most of the time, where high LET is more like the Death Star. There's no missing that. It's the equivalent of if low LET is a bullet hit on the side of a building, high LET is a 580 square meter carpet bombing of the building. Genetically, they're not the same thing. Not apples and oranges, more like apples and rhinoceros from this perspective. Another issue is that with all that DNA repair of different types occurring in the same place, they all kind of start to conflict with one another. This is a working model that we and many others have of essentially too many chefs in the kitchen spoiling the broth in this case. And we know that the biology of this, with all of these different types of DNA damage in the same place, is not effective. It's really not very good at resolving this type of DNA damage. In fact, many have speculated that we've never really evolved to cope with too much particle radiation. Sure, a little bit, but really high doses that we're seeing, for example, from radon in houses today wasn't there when we had applied selective pressure on reproduction. That contrasts, of course, to our ability to withstand high oxygen. We breathe that all the time. Infectious diseases, sunlight, ultraviolet sunlight, again, we're relatively well geared to do that, again, evolutionarily, or even alcohol. We can process and metabolize that, but essentially higher doses of alpha particles, which are really not all that high in the grand scheme of things, is essentially a type of alien DNA damage. Unless we had evolved in deep subterranean environments or sucking on the grass or in space, there would have been no selective pressure on this. So really, this is a type of alien DNA damage as far as your cells are concerned, which perhaps explains why it's so cancer-causing in people. So to understand the biological effects, we here in our labs, and of course many others pursue experiments for this, particle radiation research generally experiences somewhat of a bottleneck because traditionally it's not accessible to your standard issue biological lab. You often need cyclotrons to produce higher linear energy transfer particles. These are, of course, expensive to build, and if you build them, they have, of course, somewhat limited accessibility because, of course, everybody wants to use them. So you're lucky if you get on there a few days or a few hours a month if you live nearby one. That's not really helpful for doing a lot of repetitive low-dose exposure experiments where, for example, you must expose a cell, say, every hour, every day for multiple days in a row to reproduce kind of what's happening out there in the environment with radon. So that's a bottleneck. And if you look at the literature, there's actually almost a 10 to 1 or 100 to 1 ratio between photonic radiation research publications and particle radiation research publications, say on PubMed. We've innovated around that here at our institute. We developed an americium-based 96 well plate compatible irradiator source with a scalable bracketing system compatible with either yeast model systems, mammalian cell model systems, or whatever you're interested in to actually do this. It's adjustable dose rate, and we can get down to pretty low doses, 0.1 milligray per second if we want to, or 10 milligray per second on average. We can see the effects. These green dots represent DNA double-strand breaks or a marker of DNA double-strand breaks. In cells down the microscope in the well, you can see the field of irradiation there where we're exerting the damage. This, of course, affords us multiple opportunities to study the DNA repair pathway interplay, that too many chefs problem I talked about, or the genetics of susceptibility, knowing that we're not all equal in our ability to withstand DNA damage. A lot of our research reproduces what others have seen as well, where the rate of DNA double-strand break repair, for example, which is what you're seeing here in the two lines, DNA double-strand breaks induced by photons are repaired relatively quickly, such that nearly they're all gone by 8 to 24 hours. Versus particle-induced DNA double-strand breaks, which are exceptionally slow, with more than 40% persisting still 1 day after exposure. Completely different scenario. Again, that speaks to the complexity of what's going on here. We can do neat experiments that would be challenging to do with other technology for inducing these, as I mentioned. We can, for example, deliver 35 milligray a day, 70 milligray a day, every day for weeks and weeks and weeks, and look at the accumulating damage. Here I'm showing you DNA double-strand break markers, but of course, underneath that is all those other different types of DNA damage. We can then, of course, compare that in green there to the acute effect. So, deliver a higher dose, look a few hours later, or deliver that repetitive low dose and then look at the comparison. And if you deliver, for example, 140 milligray every day for 15 days, the cell starts to really accumulate damage more or less the same as a 1 gray acute dose in that case. We've also adapted it for yeast. So yeast biology is a very powerful model system. So here you can see increasing doses. Of course, yeast is very radio resistant. So their low doses is actually kilogray versus our own, which is more in milligray or less. But we can adapt that to clonogenic assays, as you can see here, and compare photonic versus particle radiation effects. We've studied a variety of different pathways, for example, of non-homologous end joining involved in DNA double-strand break repair, really not needed for alpha particle radiation dose we found there, contrasted, of course, to homologous recombination-related DNA repair, with which the mutants are exceptionally sick. After alpha particle exposure, speaking to the different ways that these pathways need to be repaired. We've identified, for example, mutants that are selective to alpha particle radiation. Here's a particular mutant in an enzyme which is involved in processing DNA. It's called MRE11. And if you look on the right-hand side graph, you can see there the dark green, which is the nuclease dead. It has no enzymatic activity, essentially is wild type after gamma ray exposure in terms of its ability to withstand that, versus the alpha particles on the left, which are as sick as if you knock the protein out whatsoever. So there are separate effects between alpha particle and gamma ray photonic research if you look hard enough through these things. MRE11 is a nuclease. As I say, it processes DNA through the multiple pathways that we use to repair these breaks. I'm not going into too many more details about this, but we can interrogate many of these pathways. I just showed you some stuff with the MRE11 nuclease, which helps initiate DNA repair processing. We've also looked at other enzymes. Here's the protein called Artemis. Yes, named after the Greek goddess and the protector of small children. I'll tell you why in a second. If you knock out this protein, its other name is SNM1C, you can see after photonic radiation, you get this little repair defect where about 10 to 15% of all DNA double-strand breaks persist. You can see this down the microscope, again, looking at little green dots. What I hope you can appreciate here is between 24 to 72 hours, there's a lot more green dots in the Artemis knockout cell line versus the wild type. If you, on the other hand, look at particle radiation, it's very different. In fact, it's not just 10 to 15% that persist, it's more than 40%. And in fact, if you look at 24 hours, it's more like 60% of the lesions persist relative to the wild type. This is important because this is one example of a gene that's very different across the population. In fact, Artemis, or the SNM1C gene, is mutated in a large proportion of people with Athabascan-speaking Native American heritage. So our indigenous peoples, you can see roughly where these populations are very much enriched in our nation. And there's a founder mutation there caused by a population bottleneck, which has increased susceptibility of this population to radiation, and in particular, particle radiation. So genetics is important. There's lots of these different things. Here's, for example, a DNA repair protein, another DNA repair protein, another DNA damage signaling protein that are coming up in the susceptibility factors for radon-induced, for lung cancer in general. So I'm just going to check my timing here. Good. What I want to do in the last half of my talk is to really speak about the other risk factors and then take us into low dose radiation dosimetry for radon as a question. Before I do that, I need to tell you about not the genetic risk factors, but other risk factors, including different types of houses. All of us live in probably a different house to the other person in this seminar. Some of us in single-family detached houses, some of us in apartment buildings, some of us in bungalows, some of us in multi-story, what have you. Radon is hugely modified by the built environment. Now, before I do this, I need to tell you again, remind that we measure radon in becquerels. Your typical radon test looks like this. It looks like either a little hockey puck or perhaps they're square, it looks like an ant trap. Inside those is a square of CR39 impact resistant plastic. Remember I told you at the beginning, radon moves at 15,000 kilometers per second, or the alpha particles do, sorry, from radon move at 15,000 kilometers per second. Well, that does indeed dent impact resistant plastic. That is how radon detectors measure alpha particle emanations. In houses, we never actually measure the level of the radon. We measure rather the alpha particles that they emit. You pop out that chip, etch it with a few chemicals, high resolution microscopy, spots and counts the dots. If you know the start and the end date of the test, then you can measure the becquerels per cubic meter of air, and that's how we do this. The Evict Radon National Study, led by myself and involving many others from across Canada, has been doing this for a decade. We have, this is, for example, 200,000 people and more than 75,000 properties. Each one of these dots is a Canadian house. And we've seen exceptionally high radon levels, as you can see there. We're roughly a fifth exceed 200 becquerels, and more than half exceed 100 becquerels, where again, you start to see a statistically significant increased risk of lung cancer. No wonder Canada has amongst the highest rates of lung cancer incidence and mortality in the world. We just recently published last October a summary of all of our knowledge of the past 10 years. That's at the Cross Canada Radon Survey. I've given you the URL here if you want to look at that. This has a full description of the few snippets I'm going to show you in the next few slides. In summary, though, from that, we now know that radon exposure is really a function of three core factors. Where you are, meaning the region you're in Canada, and the community type, whether it's urban or rural. The built environment, what type of building do you live in? What type of building do you work in? And then your behavior. How much time do you spend in those buildings or outside or what have you? All of these three things are the ingredients of the radon problem. Regions absolutely matter. We have a very large country, coast to coast to coast. We look at radon. The average for our nation, again, weighted by region and many other factors, is about 85 becquerels per cubic meter. Again, 17.8%, or roughly 1 in 5, exceed 200 becquerels, and a strong number exceed 100 becquerels. If you look across the regions, in this case, we grouped Canada into 5 larger aggregate regions. You can see here, for example, we go from the Pacific coast with a comparatively lower radon exposure to the rest of the country, still high globally, at 1 in 75 houses exceeding 200 becquerels, to 1 in 3 in the BC interior and Yukon, 1 in 5 in the prairies, 1 in 6 in central, meaning Ontario and Quebec, and 1 in 3 in the Atlantic provinces. These are a lot of our houses. Most Canadians have a very high risk of being in a high radon house. Communities absolutely matter between the urban to rural paradigm. This is how Statistics Canada defines urban to rural for us. A city is more than 100,000, a real rural area is less than 1,000, and then you have your small or towns, large towns in between. If we look at radon across these, and we put out a paper earlier last year on this, you can see CBC capturing me doing a funny thing there. You can see the radon levels are wildly different between the cities to the more rural areas. And indeed, rural areas have on average 32%, 31% higher radon for the same type of house, everything else being equal, versus a city. Why is that? Well, you can ask me at the end if you're interested. Buildings also matter enormously. Think across Canada, again, many diverse types of buildings in terms of age, type, and what build code they basically were subject to when they were constructed. Radon enters our houses through, of course, the medium of the built environment. Here in this little snippet, the floor there is in black, the walls are in white. That is not radon. Rather, it's a tracer gas we've injected into the soil surrounding the house. Radon partitions into soil gas. If you could see this, the entry of soil gas into your typical property, we probably wouldn't have as low radon awareness in Canada as we do today. But this illustrates kind of the effect that the built environment can have, where if you're not properly sealing the foundation, soil gas entry is very pervasive. Of course, that underlies the radon problem. The lowest levels of properties typically will have the highest level of radon, but only by about 24% or so. The next floor up is only 24% lower, and so on. And that's purely a dilution effect above. More windows and doors above ground, radon dilutes a little bit. A lot of our houses are heated by either forced air ventilation or oil-fired central heating. That really mixes the air very well in Canada, so we don't see a lot of differences by floor, although there are some. Buildings matter by type and age, of course, hugely. Here I'm comparing Canada to Sweden in terms of what the radon's like as a function of the decade that a residential house was built. You can see in the 1950s, just after World War II, Swedish houses were built with higher radon than Canadian houses. That persisted through to the 80s, where they more or less met. And after that, we diverged completely different directions. Unfortunately, Canada going in the wrong direction from our perspective. Now, having a difference of 467% higher radon for a new Canadian house relative to a new Swedish house. What did they do? What did we not do? Again, we're not sure. I have some ideas you can ask me at the end if you're interested. We've also looked at a lot of different types of houses. You can see some examples of those here. If you look at a single detached house, they will have higher radon, roughly 1 in 5 Canadian houses, versus say a row style house where it's more like 1 in 13 on average, and something in between for semi-detached. So different depending on what type of house you're in. That same effect reproduces across all communities, but it multiplies. So here you can see the trend for a city. The bungalow is king for radon. The row house is relatively lower. Both are still potentially high. In rural areas, it's the same trend, except everything is just higher. And the other two types of communities are somewhat in between those two, progressively increasing as you get more rural. There are some very highly exposed communities in Canada. Here you can see red dots is where we placed, we've looked at and monitored radon. This is all of the Cross Canada Radon Report. These are communities where 25 to 50% of the houses exceed 200 becquerels, including places very high population, for example, Regina, Winnipeg, Kelowna, Okotoks, Halifax, Nova Scotia, Kingston, Ontario, Thunder Bay, Ontario. These are communities with exceptionally high radon exposures. Now, radon is a solvable issue in a building. You can test for radon. If it's low, that's great. If it's high, you can fix it. Subslab depressurization is the technology one uses. I can tell you more about that later if you're interested, but just to suffice it to say it works. These are 100 houses in Canada, with red being the original reading, sometimes up to several thousand becquerels. Blue is the reading afterwards. Here's a real-time radon test. This house fluctuated from a few hundred to 500 becquerels, actually through the summer months of 2018. You can see exactly when they turned on the newly installed radon mitigation system. I always joke as if this was a patient, I'd be very worried. This is radon, so that flatlining is okay. There's a program in Canada called the the Canadian National Radon Proficiency Program that certifies professionals with the knowledge they need to do mitigations correctly. So we have the infrastructure to fix this problem. Right, so in the last few minutes, 10 minutes or so of my talk, and then we'll segue to lots of questions, I hope, is what we're doing with radon and people. And this is really about low-dose radiation dosimetry. To take a becquerel reading from indoor air, transfer it into a millisievert absorbed radiation dose to the lungs for a person, you have to understand two things. Of course, you need to understand how much radon is in the building. I've just finished telling you all about the work we've done to do that. The other key factor is you need to understand how much time people are spending in the building. This could be radically different across populations. We all have different lifestyles, different needs. Some of us spend no time at home. Some of us spend all of our time at home. That matters. And we've surveyed this across Canada by assessing what we're terming life activity patterns. So, for example, if you envisage the typical work from home person, or a real estate professional, or someone who works in a factory, or perhaps someone who drives truck for a living, their amount of time that they spend in their primary residence, a different residential building, a non-residential building, or outdoors or in a vehicle are all going to be radically different. But we have to understand those at the level of population in order to make sense of the radiation doses that we need to in order to do the correct molecular epidemiology for this. So there's 8,760 hours per year. That's 24 hours a day times by 365 days. So how do people split their time? This is research we published a few years ago, and basically Canadians spend about 2/3 of life in their own home. They spend another 5% in someone else's house, about 14-ish% in a non-residential building, and about 16% of life either outside or in a vehicle. This has actually changed over time in a way. We've actually monitored this from before the pandemic to today. And actually the pandemic surprisingly, due to the lockdowns, actually sent us all home. This is time spent in your primary home per year. We've sampled this in now 6,600 Canadians. And it's come back, but it's come back to a new normal where more of us are spending far more of our time at home than ever before, especially people in work. Of course, we all recognize that that could be and is work from home policies, which have been normalized. So we're spending about an extra 540 hours a year at home. That's actually a large amount of time. If you look at the amount of time we're now not spending in a residential building, that's fallen by 355 hours a year relative to the before the pandemic period. This matters because your typical non-residential building in Canada might have only a 2% risk of so-called high radon versus the 20% risk I've told you about for residential environments. In terms of the amount of time we spend in other people's houses, that's more or less back to background levels. We're spending a little less time in outdoors and vehicles. Most of this is the fact that we're not commuting as much as we used to compared, but relatively minor. If you look at the overall activity patterns for people in work versus not in work, people not in work do spend more time in their primary house. But people, again, as compared to before the pandemic, today we are spending a lot more time in our primary house. And you can compare here the sort of pie chart wheels in the lower ones, spring 24 to winter 2025, where 67% of life is spent inside for a person working, 70 to 71% for someone who's not in work. And we take that information, and because for all of these people, we absolutely know the amount of radon in their house because they did a citizen scientist radon test. This is a sample of 3,300 people in Canada. We can calculate their millisievert per year radiation dose exposures, ranging from relatively low of about 0.1 to 0.2 millisieverts per year, negligible, to this group here, which is on average 3.6-ish or 4 millisieverts per year. That's the Canadian average. That's about 3-4 times the global average, by the way, to this group here, which are receiving well in excess of 10 millisieverts a year, up to hundreds of millisieverts per year. If this was a nuclear energy worker, that would be a problem. This is from their own home. Now, not everybody will have a radon test in every home, school, and workplace they've ever lived in going back until they were a child. Now, that's problematic for a few reasons. And we actually, there's been a search, a kind of holy grail search for some time for a lifetime test for radon exposure. And this is something our teams have been pursuing for some time. Back in the 60s, there was a Nature paper that actually described the accumulation of lead-210 and polonium-210 from the radon decay chain in human tissues. They looked at a lot of bone samples there. And from that and others that followed, the idea emerged of whether we could look at radon in other more easily accessible tissue, such as keratinizing tissue, meaning your nails, on your toes, on your hands, your hair, etc. I'm not going to be easily able to get bones from participants, but toenails is something widely accessible. And we asked the question, can we correlate known indoor air radon exposure to the amount of lead-210 radioisotopes, again, that 22.5-year half-life, that accumulate in toenails? And we asked this question very specifically, is it proportional? This is something we've done over the past few years. We've controlled for everything we can think of, activity patterns, sex, age, tobacco, consumption of, for example, lead-210 rich meat for light-eating game, like an elk or a moose. Occupation, have you ever been a uranium miner? Are you a radon mitigation worker? Has your house been mitigated for radon, and so forth? This was funded in metal-free laboratories, which are just over here, here in the Charbonneau Cancer Institute, now built. That was the original artist rendition. I can confirm it does look like this, funded by CFI with a $4 million build, which we opened last year. And with these labs, we can actually get down to the femto range of detecting lead-210 radioisotopes in tissues. And it's 10 to the minus 15, or even to the high attogram range, if we do it right. And this is underway right now. Now, we did a pilot project on this funded by the Canadian Cancer Society and CHR, starting technically around 2020, but we based it on our radon testing program, which had been underway since, again, the mid-2010s. We had hundreds and hundreds of people, thousands really, for which we had radon information. And here I'm showing you a sample of the data that we used. We had a low and a high radon group in terms of household air. we knew the age of the person, we knew how long they lived in the house, and we were able to convert that to millisievert per year doses. So the low dose rate we called something around 1 millisievert per year, and the high dose rate, which ranged from 10 to hundreds of millisieverts per year for that one. And of course, we then, because we knew how long they lived in the house, we converted that to total dose exposure, invited them to provide toenail clippings for us to monitor lead-210, which we did. We had a good distribution by sex, by tobacco use history, by people who eat wild moose and elk. We monitored their activity patterns because we did this during the pandemic. We had to monitor before and after the onset so we can kind of estimate millisieverts appropriately as it changed in 2020 and 2021 to 2. And then we were able to calculate again for some people what happened when they mitigated their house. In some cases, people had lived in higher radon for an average of, say, 20 years, but they mitigated before they get sent as toenails. And so they were radon free for an average of about 2.5 up to 6 years in some cases before we saw the toenails. This was good because we wanted to see if we could still see the radioactive radon decay products. If we could, that meant we had a long-term readout of lifetime exposure. Again, we did this in the Cancer Metabolomics and Environmental Cancer Research Hub here. Took the radon level, converted it for the people who gave us toenails to activity duration, changed that to absorbed dose, got them to give us toenails, and then did the mass spectrometry. This is called inductively coupled plasma mass spec. Basically, you wash the toenails in acetone water, digest them with high acid, high heat, high pressure. It's a $50,000 microwave for that one. And then looked at all of the radioactive isotopes of lead. From lead-204 to lead-210. We do a bit of cleanup on this using natural lead-208 as a calibration control there. This is the work, I should point out, done in collaboration with atomic physicist Michael Wieser and his star postdoc Kerry Miller. What we found as a really good reading, we have down to 1 femtogram up to hundreds of femtograms of radioactive lead per gram of toenail. And we expressed all of that as a function of stable lead per gram of toenail. The ratio turned out to be important. We're all exposed to different amounts of lead. There's lead in the environment from elsewhere, and we all metabolize that differently. But by understanding the ratio of radioactive lead to stable lead, we were able to really see differences as a function of radon. So this was the outcome of that ratio from everybody. We saw exactly no differences whatsoever based on sex, based on age, based on tobacco, based on lead-210 consumption, rich meat consumption. But when you looked at radon, it really changed. There was an absolutely statistically significant difference with the low radon exposed group being very low and the high radon exposed group being exceptionally high, sometimes 300% higher or more. And importantly, there was only a minor difference for the folks that had mitigated a few years before giving us the toenails, really illustrating the bioaccumulation of this. And you can see a fairly good dose-response relationship between the amount of lead-210 and the known total absorbed dose over sometimes an average of 20 years to the lungs. Now, why is this important? And this is where I'll end before we take questions. We're doing this because lung cancer screening programs are emerging all across Canada. Just like mammography and colonoscopy, you can take the proportion of cases which are diagnosed at the end, stage 4, where we can do basically very little. Your average lifespan is 17.8 months. It's palliative. And take all of those folks and move them to stage 1, where we can, in most cases, easily treat or even cure lung cancer simply by cutting out that little nodule that you find. So this involves a low-dose CT scan. You can spot the nodules, and if you spot that nodule at that stage, you just cut it out, and that person goes along for the rest of their life. They're missing a bit of lung, but they're not dead from lung cancer in less than 2 years. Right now, lung cancer screening programs are only available on the basis of fairly heavy tobacco usage. You have to be in your 50s to your middle 70s, and you have to demonstrate a 20 to 30 year pack year smoking history within the last 15 years. That math basically means that person has a 1.5% risk of lung cancer over the next 6 years. And if you cannot adequately or reliably calculate that 1.5% risk of lung cancer over the next 6 years, you cannot be screened. We can't afford to screen everybody. We don't want to screen everybody unnecessarily. That's a bad thing. So we can't do this on the basis of radon right now, because someone just doing one radon test in one house they happen to live in right now is not sufficient evidence. But why we're doing the toenail, that could be 20 years worth of radon exposure information, and we will be exploring this to get people into trials. As mentioned before, this is the hard work from a lot of folks, citizen scientists and others in the Evict Radon National Study. It involves all of these institutions and universities from across Canada, including the good folks at CNL, Health Canada, the Lung Health Foundation, and many, many other universities here in Canada and abroad. And it's funded by a grant from the Canadian Cancer Society. It was called the Changing the Narrative Lung Cancer. It was funded in 2022. We actually didn't get the money until 2024, and it's underway right now. Well, we are right now recruiting tens of thousands of people with lung cancer. Sorry, not with lung cancer, tens of thousands of people with radon test information, either from Evict Radon or from the Canadian Partnership for Tomorrow's Health, or newcomers that come in off the street, so to speak, for us, and we'll be examining toenails over the next few years. So I see I'm at time, so I'll stop there. I'm just going to skip to my acknowledgements here, and thank a lot of the people in my team that do this. Of course, Mike Weiser and Carrie I've spoken about. That's the atomic physics team. My graduate students, you can see there in the upper left, Dr. Pearson in the top right, and a lot of some of the health economics, building science, and that done by others, including Dr. Doreen Azifa, you see there in the lower left, who's the lead of Alberta's lung tumor program. If you're interested in any of our papers, you'll find it at evictradon.org, the coming soons, and then all the published ones down below. And with that, I'm happy to take questions.
Rob, thank you very, very much for that incredible, wonderful presentation. And I think we have several questions on our panel. So we will try to moderate it and translate to you these questions. First of all, let me tell you everybody that this webinar will be recorded, and it will be available in YouTube in near future, as all of the rest of the webinar series in the past. So this will be opened to everybody in the near future. Again, so several questions around, and let me start with one of the first one. The question is, how do you isolate that the lung cancer incidents are due to radon as opposed to other factors, for example, air pollution? How you can distinguish between different factors?
The short answer to that is it's difficult right now. Lung cancer gene mutation signatures is how we do this right now for tobacco. And those are part of the tobacco signatures also associated with combustion particulates, of course. If you don't see those signatures in a tumor, it is definitely something else. There are almost half a dozen orphan lung cancer gene signatures, meaning a known signature without a known cause. One or more of those are going to be radon. We're getting really close to figuring out what those are. Some signatures are also known. So that's one way to do it. There's also the process of elimination, but also what we're going to be doing soon with the lead-210 in toenails, we'll be able to do that correlation at the level of individuals. Now, if we backtrack to populations, the population science is a little easier because you can monitor, and in fact, all of those minor studies, the residential studies I spoke about in terms of population health, can control for tobacco use. And indeed, the good studies, there's one or two that don't, but the good ones control for tobacco use. They also control for the radon, and then you can see the trends across tens of thousands of people. So that's how it's been done at the level of populations and individuals. As soon as we have those genetic signatures, there's going to be no guesswork left. We will absolutely know. And of course, then with the animal studies, we can also, in the 70s, they couldn't have gene sequenced. We can do that now. And that's one thing that we want to do with the new radon chambers that, of course, our good friends at CNL are building and should be available later this year. We will be exposing animals again to radon. We'll wait for them to get tumors just like they did in the 70s, and we'll gene sequence those tumors, you better believe. So it's been done in a variety of different ways, and even better stuff is on the way.
Thank you. We have another question. Um, you say that there is an increase around 16% in lung cancer risk per 100 becquerel for cubic meter of radon. So, what is the factor time of that? How long is the time of exposure to reach that level of increase?
13.6 years is the latency period for basically any exposure for lung cancer diagnosis. That was also very much comparable for asbestos and tobacco. Radon's no different. You need just under 1.5 decades from high exposure. And that now, mind you, that exposure might have occurred over a period of decades. But between known high exposure, the commencement of the exposure to the diagnosis, it can be an average of 13.6 years. That's in fact a very important number because that's the window of time that I need to be able to tell someone's radon exposure history through the toenail project. I actually only care about that 15 to 20-year period. Um, Earlier than that, just like giving up tobacco or you perhaps worked with asbestos in the 80s, but it's 2025. You kind of got lucky. You got lucky and you outlived your lung cancer risk in that respect. So you can outgrow your lung cancer risk with time, but 13.6 years is the latency period.
Thank you. Another question. Why do you suppose that row houses have lower radon than a semi-detached or detached house?
It's a dilution effect. So row house by nature has two walls that don't have windows and doors. They have at the front. So less windows and doors mean less points of dilution. Now, you might think that should mean it has higher radon. Well, it doesn't because now you have a much larger foundation that's split across the ground. These type of buildings also have just different air dynamics. Now, a bungalow, typically, why does it have the highest radon? It has the least dilution points relative to a very large square footage on the ground. So it's like a bigger drinking straw, right? If you increase the drinking straw, you're going to be able to drink much more Coke per second than a tiny drinking straw. So if you make the footprint on the ground, and your typical bungalow is 1.5 to 2,000 square foot versus a typical row house, which is much smaller. So those are the effects there. There are also differences by age and building code factors. Yeah, so it gets complicated, but that's one of the reasons. Yeah.
Thank you. That's an interesting question. The question is regarding this is regarding genetic susceptibility for lung cancer radon of indigenous people. Uh, the question add that the most of the current uranium miners in Canada are indigenous. This
Yeah.
is what they contend, and there is a paper describing that. So there is any literature on the increased genetic susceptibility of baseline risk for indigenous people?
Now, not specifically in the context of radon, to my knowledge. There's certainly a lot of work being done in indigenous communities about residential radon. Most of our uranium mines are not what they were in the 50s. They are ventilated or mitigated at least to the extent that any other type of mine has high radon. And it'll be a higher radon exposed population for sure. I'm unaware of literature that specifically has tied genetic susceptibility in that ethnotype for lung cancer. That said, absolutely we know about radiation risk factors that are genetic that are overrepresented in many indigenous populations. I spoke about one of them earlier, the Artemis gene, which is more heavily mutated in Athabascan indigenous peoples versus non other haplotypes. Now, that also applies to other groups as well. For example, in Vancouver area, we see a lot of non-tobacco lung cancers, but in females. Those aren't radon. In fact, that's an EGFR mutation that is very common in East Asian populations. And Vancouver area has a large East Asian ethnic genetic ancestry. And so that's why we kind of see these effects. Now, this speaks to the sort of the need for this holistic interdisciplinary science. We have to understand all of these factors, the built environment, the genetic factors, and the behavioral and the occupational in order to do this. We're certainly very interested in pursuing this for high-risk populations, which would include indigenous peoples working in uranium mines for sure.
Thank you. And this is a kind of personal question. What is the current database on northern populations and in the Nunavut province, for example? At least any data on that? Uh, for example,
So
I
in the
was
Cross Canada, yeah, in the Cross Canada Radon report, that was the one province we had a zero data on. We identified that as the area of highest priority for radon data collection. Historically, though, I'm gonna back us up a little bit. I actually did something with PBS NewsHour a couple of years ago on this. Permafrost is a very good barrier to prevent soil gas intrusion into a building. Okay, so if you have a community which is on permafrost, it typically doesn't have a radon problem. Nothing like thawed soil, so to speak. Now, of course, climate change is completely changing this equation. And there is work underway in Yukon, Northwest Territories, and Nunavut, Alaska, Greenland, and others trying to get this understood as the changing landscape of radon exposure, not just in houses, but actually in outdoor air as well, of what's going to go on as our permafrost increasingly melts with climate change. It's a very important community. There are differences in the Cross Canada Radon Report. We did have a special section on northern communities that was based on Yukon and Northwest Territories data, again, identifying the fact that we need more in Nunavut. The next iteration where we do a data update of the Cross Canada Radon Report is going to be in 2026. We have about 18 months to really get our act together. We're already working together with various partners in order to get more. If you know of people in Nunavut who would be keen to test their houses for radon, please contact me. I would love to get them a radon test.
Let me give you my experience. I was lucky enough to be on Devon Island at the North Island part of the Nunavut, actually measuring radon during
Okay.
the summer. So
In outdoor air,
internal and
internal.
external.
Okay.
So uh, so seasonal variations. You have
Yeah.
in the summer what is that the
Yeah.
frozen desert, for example. There are
Yeah.
changes in the permafrost.
Yeah.
During the winter, you have everything is frozen over there. So I assume there will be variations in the level of radon in these seasonal changes.
Yes, you would be correct in that sense. And there's actually quite a bit of data from, well, Arctic and Antarctic nations that actually speak to those changes. You can also see, it's interesting, you can change the ground and you can get fluctuations. So, for example, a seismic event is also associated because a seismic event can for sure change free phase gas migration through soil. And you can actually suddenly increase or suddenly decrease whatever radon is coming out of the soils in a given area with a seismic event. Whether natural or actually anthropogenic, for example, fracking, can actually do this. They're often temporary changes. One good example, this was in our urban to rural study last year, which we did in collaboration with architects and geoscientists, is why do rural houses have so much higher radon? It's nothing to do with the built environment per se. It's actually domestic groundwater wells. And a domestic groundwater well, which is sometimes a 50 plus meter deep into the bedrock, or not the bedrock, but the ground. You sink a pipe down. There's a gap of air around that pipe. It's called the annulus of the well. It's minimal, but as far as a free phase gas is concerned, that's a superhighway. So you actually basically gave soil gas a much faster migration pathway. And what happens is proximity to wells basically creates, because gas will do this as it comes up. This is why I was doing this in that picture. I just realized it comes up like this. And if your house is here and your well is here, your house is going to have more radon on the soil upon which it sucks when the pressure changes. That's the other one. Weather matters. So again, speaking of seasonal differences, when a high pressure system comes in, it pushes on the ground. So radon goes down. And a low pressure system comes in, it rains, it's windy. Your house will suck on the ground a lot more. So we can see big changes. This is why in Canada, all the advice from everybody, ourselves, Health Canada, everyone who knows what they're talking about here, say you have to do a 90-day or more radon test. But that's because it's doing this over the short term. You have to even that out to get long-term exposure.
Thank you. Let me go back to biology. So we have a few interesting questions. That person here is interested in inverse dose rate effect. Okay. So the question, how would you estimate the risk from exposure to radon in hotspot areas, such as those found relatively in part of Pennsylvania? This person is from the United States, but can you explain a little more what is the inverse dose rate effect and what is the relationship with the radon exposure?
Okay, so uh, when we converted from the population health studies, the 16% increase in relative risk per 100 becquerels, we actually came out with a number based on activity patterns monitored in the United States at the time and Canada, of roughly 3.68 millisieverts per year, equating to that 16% increase in relative risk. Okay, so that's the Canadian average, right? So you would set the Canadian average essentially at 16% elevated risk beyond the global population. Hotspots anywhere in the world, of course, would have higher. So you have to look at what the average millisievert per year dose exposure would be. So a hotspot, so for example, you're in a community, I'm just going to use a Canadian context, so that's where my knowledge is. I'm going to put us in Regina, Saskatchewan. 50% of the houses exceed 200 becquerels there. The average there is double the rest of the country. Now you have someone in a relatively new bungalow and they work from home. That person is going to be exceptionally exposed. They could be some of my cohort that are getting hundreds of millisieverts per year. Their risk is going to be much higher. The biology of what's going on in their lungs is going to be radically different. They're going to have that many more alpha particle events emitted from the radon they inhale and the radon decay products attached to dust in their house that they also inhale. Those are going to bioaccumulate and start hitting their cells. Biologically, then it becomes a question of their genetics. Are they resistant? Are they susceptible? Are they average? Which, of course, is a function of genetic ancestry. What happens then is going to be modified by luck. That's always a big factor in cancer. And of course, other confounding factors. Have they had a history of inflammatory lung disease? Have you had COVID pneumonia? Have you had tuberculosis? What's going on with that? And then, of course, the things that suppress risk, like their diet and whatnot. So that's kind of the big picture of what you would need to understand for a hotspot in terms of these dose rates. But it would start very much with understanding the person in terms of their activity patterns and the radon in their houses. I hope that answers your question. I don't have a better answer than that.
We have several questions. All of them compliment you for an excellent presentation. So I just will let you know people are very, very happy and excited about your presentation, Aaron. So one of the questions. This is a person who have a three-part question. The first is if you have any. Let me finish with the question and you respond if you okay, Aaron, just to go forward. So the first question, if you have any publication on radiation-induced adaptive response or hormesis, this is one. The second, they say that a few reports indicate that lowering lung cancer for the residents living in the high natural radiation background versus others. How will you comment on these studies? And the third part, do you have any suggestion for those who live in the area with high natural radiation, if you believe the lung cancer risk is high, to prevent the risk of cancer induced by radon? Should these residents take some dietary antioxidants such as sulfonamide of zinc or Rebatrol? Okay, so three parts.
Okay. Right. So first one, no, we haven't observed any hormetic effects. While I'm supportive of the background radiation of the Earth being very necessary for life, that's largely photonic radiation effects. And when we look at alpha particles, it's a completely different equation. I don't think there's going to be any dose of alpha particles because we're so genetically unprepared in terms of DNA repair for this type of DNA damage that will be beneficial. That's untrue in the case of photonic radiation. So for anyone who's thinking about linear no threshold, hormesis, threshold theory, whatever, you must, and I encourage you to separate in your mind photonic radiation from particle radiation because they're very, very different in this case. Hormesis, background radiation of the Earth, a lot of evidence coming that exists for photonic radiation. I doubt that's going to be the case for particle radiation. So that's one. Resistance susceptibility based upon areas of high natural background radiation. Same question. What is that high natural background radiation? Are those gamma rays? In which case, it's a very different set of genetic susceptibilities versus are they particle radiation that you might inhale into the lungs? A lot of these areas, for example, I am aware of some in Iran. you're going to have to understand the genetic ancestry haplotypes going on there. They could well be hardwired just, because of that environment, or just by a happy coincidence of fate, to be a bit more resilient to certain radiation types. Again, very often it's going to be photonic radiation. Will that modify risk of lung cancer? To be determined. I somewhat doubt it, because many of these studies, and there's one or two epidemiological studies, for example, that are out there that said, oh, radon might be good for you, it lowered cancer risk. When, for example, the National Research Council of Canada and other scientists, epidemiologists delved into that, it turns out that the controlling for tobacco usage was a problem within some of those conclusions. And if you don't adequately control for even tobacco usage, a population that just smokes a lot less is going to skew the math. And it actually looks like your exposure is helpful, but really that's just the fact that the control group smoked a lot less than this group. So these are the nuances. The number of those studies on that give that answer are infinitesimally small relative to the multinational dozens going back to the 70s that say the opposite. That are very well controlled for tobacco usage, as I pointed out in the slide where I reviewed all those studies. Marcelo, I forget the third question. Can you remind me, please?
Is it regarding if it is some kind of countermeasure, like a
countermeasure,
dietary antioxidants or so?
Right. Okay, so first up, I teach med students and grad students on this one. Please, everybody, be very careful with supplementing antioxidants. Cancers, if you happen to already have one that you don't know about, like antioxidants. This may seem counterintuitive. Cancers exist in an inflamed environment. Your immune system is trying to attack that cancer and get rid of it. You will disarm your immune system if you flood your body with doses of antioxidants that you wouldn't otherwise be able to achieve through your diet. So be super careful with supplements, because you don't know you have a cancer already or not, particularly if you're over age 50. Okay. Now, that said, preventing cancers, you do need antioxidants. Get those from your diet. So your spinach, eat some dark chocolate, have some nuts, some fruit, roughage in your diet, all of that kind of good stuff. Who doesn't like some dark chocolate? These are the sources where you're not going to be able to tip the antioxidant balance in your body into an artificial scenario that's going to disarm your immune system. But you will suppress reactive oxygen species generation locally. Okay, so if you are in an area of high natural background radiation, ask yourself the question, well, what is that radiation? Is that gamma rays? Well, you can't really prevent the ingress of that. If that's radon, test your house and fix it if it's high. No one needs to have a radon problem, right? That's eminently fixable. And I would argue that everybody, regardless of any of those, should try and strive for a healthy diet enriched in antioxidant producing vegetables and fruit, but be very cautious about supplementing with them. Of course, do so on the advice of your physician, but going to the store and getting super high dose stuff off the shelf, those are unregulated, uncontrolled. And in fact, I'll point to a study that was done in the United States. They did high dose vitamin E, Trolox, and a few other antioxidants for cancer patients. They had to stop the trial halfway through. The patients on high-dose antioxidants were getting more cancers, more aggressive cancers, versus the ones that were in the control group. We also have seen this in animal experiments. So please be cautious about antioxidants, but get them from your diet. That's a good thing.
Thank you. We have so many more questions. Hopefully, we have time to answer that. So let me go with another one. Very shortly, Aaron. Question is, how often should an individual from the general population test for lung cancer?
Well, you can't. You have to be referred, if you're a heavy tobacco smoker in a province with a functioning lung screening program, lung cancer screening program, you might get a referral, but your typical person cannot screen for lung cancer at this time in Canada. Only British Columbia and I think some health regions in Ontario have a functioning lung cancer screening program. Pilot projects exist in Alberta and I think Nova Scotia. There are none in Saskatchewan, are none in Manitoba. There are none in the north for this. This is an emerging screening tool. So right now you can't. And if you're not a heavy tobacco user, you definitely can't. This is one of the things we're doing right now. So sorry. But you can test your house for radon and fix it if it's high. So that's one way you can sort of change the roll of the dice for that one.
Yeah, these are a few questions regarding the how you test for radon. What are the technologies available? How expansive should be that detection system across Canada on the houses?
Mhm.
So can you
So
briefly comment on that?
yep. Okay. So I showed you the little hockey pucks. That's they call an alpha track detector. That has the square of CR39 plastic. Those are relatively inexpensive. You're looking at something, depending on who you get it from, the low 50s to the high 60s dollars. So that's fine. The evict rate on national study, again, citizen scientist, we do these at cost. So again, disclaimer, I make $0 from any of this. It's all just done through the research program. I think they're still $54 right now for that. That's the lowest in Canada. They can go all the way up. Now that's for an alpha track test. There are digital tests. And I showed you some real-time radon data when I showed you my patient crashing out. That was the house. That's a real-time digital test. Now those are going to run you out of zero. You're going to cost you about $500, not $50 for those. Some are great and some are hot garbage. In fact, Health Canada is about to do a recall of a whole bunch of fake devices that are for sale on Amazon.com. So I would encourage you, wherever you get your radon test device, please don't get it from someone you don't trust. Get it from a research program like ours, which works with CNRPP professionals, or get it from the CNRPP professionals, Canadian National Radon Proficiency Program. It's a Health Canada regulated certification. If you have a problem, you can complain to CNRPP. They will discipline the mitigator. I know a lot of them. Again, no financial relationship with any of them, but they know what they're talking about, and they'll quality control your test. If you look at the Cross Canada Radon report, one of the things we had early on are all the quality controls that have to be done for this. So 5% of, for example, of all people in the Evict Radon National Study get a duplicate at random at no extra cost, right? So we always have duplicates. We want to check on the precision. This, how well does one test reflect the same test right next door? We also do all of the spikes and blanks to measure accuracy. All of that's in the report. You can view that if you want. So testing, roughly 54 to high 60s for an alpha track test, $500 if you want the fancy digital devices. Get those from someone you trust. Now, mitigation, I know that's going to be another question, so I'm going to preemptively answer that one. There are about 200 professional Canadian or CNRPP certified professionals in Canada at this time. In the rural areas, it can be a little harder to get them to come out, and it will be a little bit more expensive because they incur travel costs. But if you're in an urban area and you've got a mitigator near you, your typical costs are something between 2.5 to 3.5 thousand for a typical mitigation. It can be cheaper if it's a super easy mitigation. It can be a little bit more expensive if it's a harder mitigation. Like anything in HVAC, you don't know what it'll cost until you diagnose the house. It's a 2-day visit. You're going to have a diagnostician come in. They're going to do some pressure changes, figure out what they need to build a system, and then 1 day to fix it. And then that building is permanently fixed for the rest of its lifetime as long as the fan's working. So this is not something that you have to keep doing. I always joke with people, if as a cancer researcher, if I could sell you a pill for $500 or $5,000 and you never have to eat a vegetable that you didn't like or exercise again if you didn't want to, but you'd have all the benefits from a perfect diet and a perfect exercise regime for the rest of your life. Would you take it? I would, because I could be super lazy for the rest of my life and get away with it. Now, radon is the same. You can get a radon mitigation for where you live, and you never have to worry about radon-induced lung cancer again, unlike the 3,000 Canadians each year who do.
Thank you, Ron. There's an interesting question. Is regarding the toenails methodology.
Yeah.
How is possible that you can measure 15, 20 years of dose history uh, in toenails? Because the toenails are in places every 12, 18 months. How you do that?
Okay. So now we're in lead metabolism land. All right. So lead. Why is lead so poison and toxic? We bioaccumulate this in our bodies, right? So it's sequestered in our body. So of all lead that we absorb, whether it's radioisotopic or stable lead, 75% is retained over decades, but at any given time, 25% is being steadily cleared through a variety of tissues. Hair, nails, skin, it ends up in our chubby bits, so to speak. It ends up in our bones. Okay, now, why can I see 20 years well, we think we're going to be able to see at least a decade. Hopefully we get to my 20 years. I don't know yet. That's what the validation trial is about. But we can at least see past the 5 to 10 year mark. That's because all of this lead, which we're, in this case, radioactive lead absorbed in the lungs, is being sort of sequestered elsewhere, but then slowly our body is getting rid of it. And that's what manifests on a regular basis in any of our tissues. And so, yes, actually, the growth rate of your fingernails is about twice as fast as your toenails. Your toenails, your typical Canadian, I know this because we have data on it. This is weird. Most people are cutting their toenails every 30 days. They're cutting their fingernails every 15 days. Men grow slightly faster than women. Men cut more than women. But that's a short timeline. But importantly, it's your body, which has sequestered it, which is constantly trying to get rid of it. So it's an excretion pathway, essentially. That's why we can see long-term exposure with this, what's coming out, in this case, by toenails. Yeah.
Thank you. Well, we have 5 minutes. Let's see if we can put a couple of questions before we end.
Okay, I'll go quick. Go quick.
Yes, there's a person interested in what's going on in terms of radon measurement related to the Canadian forces, our military forces.
Right.
In particular,
So short answer: in the late 2000s, all federal buildings, which included all military bases, were tested for radon and mitigated if they were high. If you're on a military base, you're probably safe. Next question.
Excellent. The lung cancer risks are compared between male and females. Females are more susceptible than males.
Yeah, there's some evidence for that. We don't quite understand it. It's been long known that non-tobacco lung cancer is more prevalent in females. Some of that is in outside of Canada, and for example, a lot of countries where solid cooking fuel, and females are more likely to do the cooking fuel, that exposure. But there are some sex-based differences that we're trying to understand that. I don't have an answer beyond that, but yes, there appears to be some bias between the sexes. One example, for example, that we know about, which is behavioral, is that still today, women are far more likely to spend more time in the home than males. Part of that is childcare.
Okay. And I think that the final, I think it's a comment regarding radon in miners. It looks like the main study come from Port Hope uranium processing workers.
There's lots. There's lots of studies, but yes, yeah, they're among them.
That this person recommend use El Dorado study from Lane and collaborated in 2010.
Okay.
Yeah, but that's a recommendation. But I think if we have more questions, I would try to pass to you, Aaron, so we can answer and pass to the authors of the questions later on.
I would also say I have a whole team of people that can answer radon questions. You just have to email info@evictradon.org. And if the question needs to come to me, it will come to me. If you have questions about radon testing, outcomes of the Cross Canada report, PhD scientists who will answer your questions as well, so we can answer everybody as they need it.
Excellent. Well, Aaron, has been a fascinating lecture. I appreciate so much, and I think the people are very excited about this, and we learn more about the radon issues. And in particular, to me, it's very interesting because I'm interested in space radiation, and high altitude radiation low is a passion of mine, and is a quite different. As you say before, it's not apple and orange. It's an orange versus a dinosaur or the elephant.
Yeah, I say rhinoceros. Yeah.
Yeah. So it's fantastic. Good. Thank you very much. I would like to thanks to all the participants and the audience. Please forward questions to Dr. Goodarzi. And we can repost again your email address or your directions around for the general public. So Aaron, if you can stay with us a little more, we'll do a debriefing. And I want to thank everybody to join us. The low dose research webinars is stable on us here in CNL, and we will continue. And so we will advertise in our next speaker, and hopefully you will join us again. Again, thank you very much for your participation. Thank you.